Okay, nice. Thank you. So welcome all participants once again. I can see there are now 79 people who have joined in this session. I welcome Professor Nirala Ramchiary, who is the speaker of this session. Dr. Ramchiary received his PhD from Department of Genetics, Delhi University. He is currently working at the School of Life Sciences, Jawaharlal Nehru University, New Delhi. After his PhD, he pursued a very productive postdoctoral research at Chungnam National University, Daejeon, South Korea. And I was very fortunate to visit him during those days and meet his professor and mentor, Professor Young-Pyo Lim, who has become a very close friend. With Professor Lim, he worked on the genomics and genetics of brassica crops, including Brassica rapa genome sequencing. Dr. Ramchiary's laboratory is actively working towards understanding the fiery hot pungency development in Bhut Jolokia, which is called as the ghost chili from Assam and adjoining areas like Nagaland and Manipur, where they are known by other names like king chili and so on. And what are the effects of this particular chili, which was once declared to be the hottest chili in the world, on human health? So those are some interesting areas of his work. His other research interest includes study of nutritional health, beneficial compounds of wild vegetables, and medicinal plants of Northeast India. He has published several reputed in reputed journals of international publishing houses. Currently, Dr. Ramchiary is teaching genetics, crop genomics, biodiversity and evolution, and computational biology to postgraduate students. So I now request Dr. Ramchiary to deliver his lecture to the participants, and kindly please upload your presentation.
Yeah.
Yeah.
Am I audible?
You are audible, but the voice is somehow low.
Still low? Still low? It's fine? Hello? Hello? Hello?
Yes, yes. Yes,
Okay.
we can hear you.
Yeah. Thank you, Professor Bora. And I'm really thankful for you to for inviting me in this very nice, FDP. And first of all, I apologize for yesterday's problem due to sudden medical emergency. I couldn't, do as per scheduled time. So today again, nevertheless, I'm presenting the the same topic.
Yeah,
Yesterday.
Dr. Ramchiary, you may switch off your video and then upload, and then probably the voice will be better. Yeah. Kindly upload your presentation. Yeah.
Yeah. So thank you once again for inviting me. And
so there is a suggestion: you may have to hold your mic near your mouth. Sir, please try to keep the mic near the mouth. Jagadish is writing. So you may have to hold your microphone.
Okay, okay. Now it's okay, clear?
It is clear now, yes.
Okay, okay. Thank you so much. Thank you so much.
Welcome.
Okay,
you have to thank Hema Jagadishan. Yeah.
Okay.
Yes.
Thank you, Hema. And
Dr. Ramchiary, just a little information. Maybe you can take small breaks after 20 minutes, 30 minutes, 40 minutes, whenever you feel like, because that also helps the participants cope up.
Okay.
Yeah. Thank you.
Okay. Thank you once again. Good morning to everybody. And today's topic is genome engineering for environmental applications. And you know that what is we are going through. We have climate change. There is no water, and soils are degraded. And if you see the farmers are crying because of soil are deposited with salt, as well as different pesticides, as well as mechanization of some of the soil. That's why crops cannot be grown. That has to be taken care of later on with the help of biotechnology or the plants that we are growing, or any other mechanism we have. Because of this, we have hunger. Everywhere there is hunger, especially if you look at hunger index is very high. And if you look at the forest area, all because of climate change, soil erosion, and all the plants are dying. Still, these earlier forest lands are showing to be barren land now. If you look at here, soils are being eroded, and if you look at the farmers now, excessive use of pesticides or fertilizers, they are all plant fertilizer or growing of crop plants in the particular soils is being becoming difficult. On the other hand, we have, if you see this one coronavirus, many viruses, on many plant diseases, which is occurring in the farmers field. That's why with the change of climate, or with the rainfall pattern, temperature, or cold, and every other changing climatic conditions, this diseases, virus, bacteria, all affecting the human health as well as plants or animals, whatever it may be. Then if you look at that, down here, we find that there is pollution because of emission of carbon dioxide is more, methane is more. That's why we have global warming going by. And here, the last row, if you look at, there's lots of pollution. We have lots of plastics. We have lots of other material that are being polluted still, and then this also causing the losing of biodiversity in this particular. Now, when you say that, can you reclaim the soil? Can you find some biological material or biological organism which can degrade these plastics, or which can control, or any mechanism that we can biologically control these diseases, or we can reclaim this soil to be more productive by looking for more of plants or any organisms, and bringing back to normal soil condition? Or can you reduce the pollution by using any of the biological material that we have in the world? So looking into this title, and it will deal with the biological material today. And if you look at the terrestrial food chain, we find that there's producer, then small herbivorous mammals, which first feed on dead, and last predator where any small mammals produce. All this makes a food chain, and there are different levels of organization of different levels of organism, and it helps in recycling of the nutrient or chemicals or organic material in this on this earth, on the in the water, in the environment. That's we have to look for those now. Looking at the biological material, as all biologies organisms are involved in recycling of the nutrients and recycling of the soil, recycling of can you degradation of plastic, or can you produce the biofuel based on that. Whatever living organisms we have, nature has given so much to us that we can find one or the other, for all the purposes that we are looking for here. And remember that these all organisms contains, DNA, which is nothing but genetic material which transmits from one generation to another generation. So we're looking at you have gone through the last few lectures that every organism has have DNA. The DNA and help in continue the generation, the same organism, the next generation. And if you look at that, in the nucleus or chromosome, DNA will be constituting like this double helix. And when this fragment of DNA produces through transcriptome RNA polymerase, it produces RNA, and this RNA may be producing protein, which is which expresses and particular phenotype, governs particular phenotype, or it may not be producing protein, but it may help in regulation of the biological functions in organisms. So whatever it may be, segment of DNA from here, if you produce it, we call it as a gene. To be able to engineer any of the genetic material, we need to understand what is the genetic constitution in this particular genome. And you know that what is a genome? It is a sum total of DNA element of DNA present in any of the haploid organism in a diploid organism, because we get one copy from father, one copy from mother. So that is what happened. We only take the one copy each from in a human being, except for the Y and X chromosome in case of human being, because Y chromosome is belongs to male, X chromosome belongs to both the male and female. That's how we say that there are we have 23 plus 1 pair of chromosome in a human being. So again, this is known as the central dogma, which I'm sure that you must have gone through the earlier presentation that when the segment of DNA out of this long DNA it produces transcriptome, then RNA is produced, then it may produce protein or may not be produced protein. But nevertheless, if it doesn't produce code for amino acid or it doesn't code for any of the protein, we say still it is a gene because it has a regulatory role, like we have transfer RNA, ribosomal RNA, or nowadays there are researchers are reporting lots of non-coding RNA, and we know that out of the total 3 billion base pair in human being, 90%, 95 to 90% human genome is found to be producing RNA, but only 1 to 2% of the genome is producing protein. That's why people are saying that around 95 to 96% of RNA produces, which is produced in human body, is not the coding or translated to protein, but they have some other regulatory role. So now for genome editing, first of all, we need to find a sequence structure of the gene or the genome. When part of genome which is present in chromosome, it produces a gene because it produces a whole set of DNA nucleotide that are present in the chromosome may not be producing RNA at the same time. That's why we have to find two ways. One is you have to find sequence or structure. Second is you have to find function. Now, if we now there are so many model organism people have done genome sequencing and they have predicted based on the previously reported genome sequence of related organism that this may be this particular sequence of segment of sequence of DNA in this related organism may be coding for this particular gene, which is known as the annotation. So to get earlier, what people did was that we first screened genetically, where in case of plants or in case of Drosophila, people try to understand, for example, eye color, earliest example, T.H. Morgan, that found that particular chromosomal segment or morphology is linked to particular phenotypic trait. But it doesn't create, at a large scale analysis, we need to have many more at a time analysis, protocol where you can analyze large number of DNA. So it came with the advancement of DNA technology, DNA sequencing technology. There are lots of nowadays you can sequence a human genome or any of the plant genome or any of the microbe genome in a very shorter span of time, along with the investment of less money. Not only that genome sequencing structure is required, we need to transcriptome. Out of these, that for example, 20,000 human genome, 20,000, 3 billion human genome codes for only 20,000 genes, protein coding genes in human. And we have to understand that out of 20,000 human gene, which is producing in which tissue, because it is literally developmental specific, we have difference in expression in our different tissues, organ. That's why we have to find out what gene, what tissues are expressing what, except for some of the housekeeping genes, we find, there are set of genes which are expressing particular tissue. Now, only that RNA, as I already mentioned in previous slide, that not only the RNA, because RNA may not be, producing, all RNA may not be producing proteins. So we have to find a protein, because ultimately protein is produced and it gives a phenotype. But nevertheless, that RNA which doesn't produce protein, it also regulates the genes which produces protein. That's why we say that regulatory RNA or protein coding mRNA. Then we can find whole genome, or in case of nowadays, diseases, we find that there are lots of microbes, and in your soil, your water contamination, we have our human body or any other, place or habitat you take, you get lots of thousands of millions of, microorganisms inhabited, which may be useful for us or which may be causing diseases. For example, nowadays, if you look at coronavirus, oily spread. So we have to find a DNA structure, then RNA, what RNAs are producing, then we have go to protein, that it helps in different proteins are proteins different phenotype, and there what are the different base pairs, because we know that there are only in eukaryotes we have only 4 nucleotide, which is combining different permutation and combination and producing different genome size and different genes. Although we have only 4 nucleotide base pair in universally in eukaryotes. So again, then nowadays we know that if finding the RNA or finding the DNA structure of protein, it doesn't give you perfect function until unless you know that we have to correlate with the phenotypic expression that are being seen in particular individual of an organism. Then only we can correlate. If you see the phenotype, we can find that there may be something wrong in the genetic makeup. Or if you look at the uh, you know genetic, you dissect the the genetic sequences or gene sequences, then you mutate it. If you see the phenotype that there is some role in this, that's why we say that looking for from phenotype to genotype, we say forward genetics, as well as and a DNA sequence or function of the gene to phenotype, we say the reverse genetic. Nowadays it's being famous. But why I'm saying this, because we need to understand that when you say that genome engineering or gene editing, whatever, or genetic engineering, we say that we need to get the gene sequence or segment of the genome which can be edited or which can be is it clear now? So first thing is that we need to understand that what are the genes that are producing particular phenotypic trait. Now, if you look at different literature, it has been shown and defined by many scientists that what is genome editing? What is the difference with the earlier conventional genetic engineering? We say earlier, is it that genome editing and genetic engineering are the same? That we can say sometimes we say that genome engineering, sometimes we say that editing. Sometimes here only different is that we can edit, we can insert, we can replace, or you can remove particular segment of DNA or gene. That's why we say that gene editing or genome editing. But even though genome editing is being largely used, but people are mostly using the one or two gene. They are doing the one or two gene editing or replacing or inserting or creating mutation. But here only the advantage here is that we know the side of the insertion of the particular foreign gene or whatever you may be inserting, or DNA fragment that you are putting into. But in case of earlier like agrobacterium or biologistic methods, where there are so many methods of putting a foreign gene or gene to particular organism, for example plants, we don't know the exact location of the genes. That's why mostly we say that it is the site specific editing, but there we don't know. That's why there is a major difference between these two, genome editing or genetic engineering. So there are several technology that have been being produced for genome editing, that earlier zinc finger nucleases, where we'll be looking into the next slide. We have transcriptome activator like effector nucleases, RNA guided endonucleases, and multiplex genome editing by CRISPR case. And out of these many, due to different advantages, CRISPR-Cas9 is being widely used because of easy and easy to use, and success rate is more. So if you look at, and you might have read, you might have come to know different, different methods, like from your from the time schedule. I have seen that this Italian zinc finger motifs presentation has been done. Nevertheless, I'm just briefing in this one that it is a CRISPR-Cas is a newer technology, as you know, that we like. It is a site specific. We need to have small guide RNA, which is around 20 nucleotide long. Then what it 20 nucleotide long. It takes the Cas9 nuclease along the PAM proto. protospace associated motif, then you go there nearby this one. So we find that there is site specific. Here also the talents also we have site specific recombination when there is nearby the sequence complementary is there. Then in between this left and right we have zinc finger. Then there is cutting of the DNA segment, this double-stranded break that are being created. All this meganuclear single finger motif talents and Cas9, they produce a double-strand break, which we can have two kind of joining homologous recombination where we need to insert some of the gene, then we add some of the gene, then we can do go for homologous recombination. But if you want to mutate in the gene or particular genome, we can inactivation or joining of non-homologous and joining will be there. So different methods have different advantages and disadvantages. Here, that is I think the slide is very small. But you can see, I think if you're looking at here, origin, if you zinc finger motif, eukaryotes, talent is bacteria, CRISPR is bacteria, archaea, structure is dimer monomer. Here we say that these are the three different methods that are being used, and this is the comparison. Design simplicity. If you look at zinc finger, nucleus, then we say that it is. They have found that it is moderate in terms of design simplicity and slightly complex talent, and CRISPR is simpler than any other these two methods that are being used. Then engineering feasibility, affordability, low, limited, moderate. Here it is high. This affordability is very high in case of CRISPR case. Popularity, affordability, it is very high. DNA binding molecular recognition mechanism. It is we have J finger protein interaction at introduce double stranded bread. Here also double stranded. Here with double bread. But we need here in case of CRISPR case, we have CRISPR RNA as well as small guided RNA along with the which creates the Cas9 along with creates double stranded break. So modification pattern is like this is FOG1 nucleus is there, FOG1 nucleus. Here is Cas9. Nowadays there are so many cases that has been that has been discovered, Cas12, Cas13, or something like that's what. And specificity determining line of recognition site. That there are typically 98 base pair. Here in case of CRISPR, it is 22 base pair. This one. These are some of the advantages of CRISPR who compared to the gene figure nucleotide and talent. So overall, if you look at that, this is CRISPR is being widely used because of easiness, less accuracy is more compared to any other these two methods being used. So now you know that we have pollution, we have carbon dioxide availability, we have so many things, chemicals exposed surrounding us, and that's why we get our body also get exposure to those chemicals and environmental hazards. That's why we get diseases sometimes for so many diseases. For example, due to environmental toxicity, we may have paraquat as a herbicide, which when exposed to this particular herbicide chemical for a long time, we get Parkinson's disease may develop, generates reactive oxygen species, which causes cell death. So people are looking at what causes exercise. As a biotechnologist, we need to look for mechanism inside our body. What gene is responsible for receiving this paraquat in our body, because there has to be signal, and that chemical has to go inside our body, which signals regulation of expression of some of the genes, and ultimately cell cell division or mechanism inside the cell. Normal mechanism may be hampered, because of which we get diseases. So paraquat being used widely by farmers, it has been found that whoever associated with paraquat, chemical is being, found to have Parkinson's disease, and it's also causes cell death because of generation of lots of reactive oxygen species. So they have, people have done that to find out can you use any of the molecular mechanism which can prevent that Parkinson's disease when exposed to paraquat or this particular chemical. So by understanding that this by expression analysis, it was shown that this particular gene, three particular genes like cytochrome P40 oxidoreductase, ATP7A copper transporter gene, as well as SL, SLLC45 A4 sucrose transpose gene is found to be highly expressed and which ultimately causing the paraquat induced cell death. So it is from their expression data, because we already discussed that we need to understand what particular environment or hazardous condition that set of genes or particular gene is being highly expressed, which may be creating the abnormality in the cellular mechanism. So these Ragar and et al. They have found that they have reported that in their review that because of these three are being highly expressed, it is likely that there's signaling of these three genes are making another gene express, and that's why the paraquat induced cell that is being found. That's why they have screened they have screened with a CRISPR-based experiment and found that found that uh, found that these three transporter, if block expression of these three genes is blocked, then there will be less effect of paraquat in the human body. Furthermore, they have reported that this POR, which is cytochrome 450 oxidoreductase, is a source of paraquat-induced, reactive oxygen species production. So this is how we can use by looking into the gene expression, how this environmental toxicity can be reduced by if you can modify, unless and until if it doesn't drastically affect the function of the normal functioning of the body. So now for environmental toxicity, we know that we need to understand that what is the effect in the human body. Suppose you get the exposure to environmental contaminants, chemical inhibition is there, then mechanistic events underlying disease. What are the cellular mechanism? What genes are expressed? What proteins are expressed? That we need to understand. Then only we can go for silencing, knockdown, or editing of the particular gene, so that it prevents this occurrence of particular default or abnormal mechanism. So we have an RNAi mechanism or technology where we just reverse sequence of gene is being introduced in the in the in a genome as a talent that recently produced, genome editing technology, CRISPR-Cas9, as we know that we have been discussing from the last previous slide, that it may alter receptor-mediated signaling, because everything that we get signal, our skin or our membrane has proteins, which those proteins accept the signal, and those proteins transfer the signal to another inside the cellular compartment, and then it induces some other genes to, whether it induces to express some other gene, and it may cause cell death, it may cause cell proliferation, or it may cause DNA damage, or it may cause inflammation or oxidative stress. That's why we have to understand here that we, by exposing particular tissue or body in vivo, in vitro, we need to understand what gene is being expressed, and shortlist those genes, and design editing a cassette or gene silencing cassette, so it CRISPR case or any talents or RNAi, then we can silence and see how it works in this one. So we have now what should be the strategic concept? It should be to understand that how to go for this, understanding or finding out the molecular mechanism inside the body. And can you hear? Still not able to hear? You can hear or yes? No, no. Okay. Okay. Thank you. So we need to understand that by what strategy we have to use relevant and relevant to environmental health, hazardous chemicals, mechanism of action, adverse outcome, biosynthetic pathway or metabolic pathway inside our body. Then, if we need to identify specific genes or key genes involved in eliciting toxicity or susceptibility of that one, or determination of potential chemical toxicity across screening methods, and population response variability, where we get that out of 100 individual or 1,000 individual, if you look this particular chemical, some may be highly susceptible, some may not be highly susceptible due to the genetic differences in the genome that we have to understand and try to understand and use this information for further designing of editing of the genome or treatment of biological treatment from molecular methods. Then public health solution, reversal and prevention of contaminated into diseases that can be done with the help of, if you know that all information at hand, then we can design available methods to control the one. Now look at this one, that diseases due to environmental effect. If you go out, we have been in a lockdown since the last 1.5 year because of fear and that this disease is highly contaminated in the environment. It may be in the air, or it may be anywhere. That's why we are isolating ourselves inside home. That if you look at India condition, we have we have 3 crore 17 lakh 54,281 people have been infected. Total cases, this has been discharged. There is 4 lakh 35,758. This is being this data is I got today from the MyGov in .covid-19. But if you look at the world, that there are so many people died, and this is 44 lakh 74,301. This is all the environmental created. It has spread so much that how to control this one, and sequencing of this viral gene is found to be 30 kb around with many URF, and people try to control this virus by vaccine, or by can you design some RNA or CRISPR case cassette where we can insert, or inside our body, or you can take, then can it be silenced, or that symptom can be stopped, that people are trying. And this is the first report that I published in Cell, that development of CRISPR as an antiviral strategy to combat SARS-CoV-19 influenza. They've designed CRISPR technology by taking the sequences around 1,000 SARS-CoV-2 or coronavirus being available deposited in the database. They have bioinformatically aligned together and found around 40 conserved sequences among those 1,000 viruses. They have analyzed and found to be conserved sequences, and they look for the CRISPR site and designed guide RNAs where they can silence and try to experimentally in the human lung epithelial tissues. And they found that out of these six, around 40, 42, guide RNA or CRISPR cassette they have designed, only six are found to be around 90% of the, 90% of coronavirus strains they found to be similar, and which can, they propose that the six, RNA CRISPR, CRISPR-Cas design can be used for the silencing of 90% of the strains reported till now. So they have found this in the experimental experiment that the influenza can be silenced totally, or it can the virus can be cannot survive if you use this guide RNA and it being degraded in the lung epithelial tissues. So this is what what is the profilated antiviral CRISPR in human they have designed, that is CRISPR for pan coronavirus protection. They arrange multiple virus strain alignment they have done. They have identified conserved regions here, and they found that these are the some of the conserve, and they found several conserved areas like this. And this conserve and CRISPR RNA validation and cell culture. This is a nutshell. They have said that this conserve sequences being CRISPR RNA being designed to introduce in the lung epithelial tissues, and then virus challenge, and they have infected with virus. They found that, and influenza virus is not there, and they couldn't multiply in case of lung epithelial tissues. But due to in human they couldn't find live coronavirus, that's why they couldn't introduce. But nevertheless, they say that if we can develop some of the safe introduction of coronavirus or any of the virus inside the respiratory tract, we can find that whether it can be used to silence or suppress the coronavirus inside your body. So what they did is that this is a simple mechanism how the virus infect the human body or cell. That this is the virus, it goes inside the cell, viral entry, then viral releases what it's RNA, genomic RNA, it multiplies translational protein, capsid protein, envelope protein. What will be coding here, then replication will be there. Then what will happen, the new virus genome will be multiplying, and we have new viruses, and then giving symptom to us. But what we can do is that while we can silence the receptor here, and designed about those best conserve CRISPR sequences. They have designed and found that if you can introduce this in the cell, it silence or degrade or clip down the viral RNA. That's why we may not get the symptom in the cell or in vivo. So they have done, this is the alignment of this is the viral genome, coronavirus genome, where we have around 29,900 something base pairs that they have taken nucleoprotein as well as RNA directed RNA polymerase, because these are involved in the multiplication of the virus. They designed this one, and they have done infection in the lung epithelial tissues. They found that gene expression, they found within 24 to 48 hours, they found that virus, influenza virus, is not there. Now, if you look at here, this is more convenient compared to the our RT-PCR, because of RT-PCR take lots of time. That in this slide they have shown that these 6 CRISPR RNA that are being designed based on the alignment of around more than 1,000 corona strains are seems to be highly effective. So they have found that Cas13d PEG man is here. Remember that corona is RNA virus. That's why Cas9 cannot be used. But Cas13d can be used for cutting or degradation of the nucleus, can be used for degradation of the DNA of the virus. So they have used this instead of Cas9 nucleus. So it is able to inhibit influenza A virus infection in human lung epithelial tissues, thereby showing potential for the SARS-CoV-2. This is the first application and highly timely application of the CRISPR in technology to silence the coronavirus but still it has to be tested because they couldn't do in the live coronavirus because of deadly, infection. So likewise, if you look at the genome editing, we find that as a viral, we find that in virus, what we find is that, if any of the virus infect here, there will be receptor, and mostly that nearby membrane, we need to, silence if these viral receptors, which, bring the virus inside the body, is proteins are not there, then maybe entering of the virus DNA or RNA may not be possible. But if it is inside there also, we can design, CRISPR RNA, which cuts the after recognizing the sequences, sequence specific cutting of the virus foreign RNA. Then we can design this kind of methods through genome engineer or gene engineer or gene editing, or we can silence then one. Then we may not get a virus disease. That's why. So this is the recent studies of genome editing therapy of viral infection diseases, because viruses are widely highly found in the environment. That's why people are trying to understand all the genomes, because virus is very small. They can be sequenced and try to understand what is the sequence composition of a virus. As are there virulent genes, and since we know that CRISPR can be used for silencing of those DNA or RNA of the virus, then we can design based on the sequence specificity of the viral genome, and we design a primer. For example, here HIV, HIV for HIV CD4 T cells in people, the zinc finger nucleotide that method was used to silence the that CCR5 receptor of the HIV, and these are some of the example where that genome editing or gene rather we said that gene editing technology or therapy being tried in against the viruses, but it has to come to people's use, because if you look at, and even though CRISPR has been, or genome editing technology has been reported 20-30 years back, but still we have to use, but people are giving more of CRISPR, technology in virus, in bacteria, in plants and animals, or whatever maybe biological system that we observe today. We can use CRISPR, technology to do edit the genes, or add some new trait, or modify the trait, or phenotype, or some degrade the gene, or mutate the gene, something. All we can use the CRISPR. That's why people are being used. So we have different human hepatitis B virus also, and many people have done. They have designed CRISPR case, but saying that we need to prove of concept. This is the one we need to take the from laboratory to bedside. If you can take this whole technology and silencing of the since viruses are very highly mutable, and they have multiple, their multiplication very higher rate. That's why every time you design a particular, CRISPR silencing, cassette, it may not be useful, because the next generation after few hours the virus may mutate. That's why this study, previous study, they tried to find out the most conserved around 40 to 42, CRISPR site, so that they can design and silence whenever it is in large scale in many viruses. So genome editing techniques promise new vectors of water, environment, microbial technology also. That recently this review has been published that environmental remediation we can use by using the CRISPR technology or genome editing technology, any other genome editing technology, because we know the environmental bioremediation is can be done by either by microbe, algae, or plants, rather than higher eukaryotes, like animals. So if you remember, there is I have not taken here in the slide that in 1999 or something like that in National University of Singapore there was one zebrafish being transferred that jellyfish green fluorescence protein, and it was first being used for to understand, to trace the water pollution. But later on it has been, there are different color zebrafish being developed, and it being used as the ornamental fish around the world, including the US and Singapore and the other. Uh, Although initially it was used for the monitoring the water pollution. So we have, as you know, that drinking water and whatever water we have, pathogen detection is there, as well as we have some of the microbes that are being found to be resistant to antibiotics, that also found. And we have lots of pollution in the water, more of carbon dioxide is there. And can you utilize, identify some of those organisms like algae or bacteria, which can use this carbon dioxide metabolism, and it can clean up the water body. As well as you have wastewater treatment, we can use with microbes nowadays. Although we are using most of the with the chemicals, but biological system like algae and bacteria can be used for treatment of the microbe. There researchers around the world are being engaged to identify what other microorganisms or organisms or algae that are found in the contaminated site, and can you use those genes or some of the genes or those organisms itself by modifying their genetic makeup to clean the wastewater or carbon dioxide or pathogen or any other thing. That's why we have uh, these people, Wu and his colleagues, they have used Aeromonas hydrophila that is involved in the bacteria that is found mostly in the pollutant areas. They can transport and so they can metabolite, you know, uh, uh, they can degrade uh, contaminated water or soil wherever they are there. Like they have done with CRISPR technology, they have identified genes. This is FTZ genes quickly, accurately. They have identified by CRISPR technology that this gene is involved in remediation of bioremediation of water body, contamination of water body, as well as they found that ARS reported that ARS operand played an essential role in arsenic detoxification. We know that what is the operand? A cluster of genes is being, transcribed or being RNA is produced from a particular promoter, and that's why very prevalent in bacteria. So we say it is an operand, and it was found that arsenic operand, the genes which is responsible for arsenic detoxification, is required for in this particular bacteria. And if they try to understand by silencing, can we do something that or metabolizing more of the or removal of the arsenic from the water body, and they found they could be successful. So again, water that Salmonella oneidensis is also bacteria. They found the extracellular respiratory bacteria. They've taken 10 genes to silence. If we can 10 genes are being silenced, how this metabolizing of the chemical in the environment or pollutant degradation is being done. And out of 10 genes, they have found that 7 genes are involved. If they are silenced, they can clean the they can degrade or they can help in degrading the pollutant in the soil. That is by using the CRISPR technology only. So CRISPR being used by many of the technology. Can we have a rest? 5 minutes? Yes. Okay, 5 minutes, please. Can we start now? Okay. So CRISPR can be used for understanding the mechanism of pollutant degradation in bacteria. They have used from previous two slides that we have seen by two groups. Now here it's very widely known that malaria is carrier that Anopheles is carrier of malaria pathogen parasite. So this Dong and his colleagues in 2018 they have reported that but if we silence through FRP1 using FRP gene which is fibrinogen related protein 1 by using CRISPR technology. It has found that it found that there is no malaria parasite was found inside the insect, and when this insect bite to human being, there will be no transfer of any of the malaria parasite in human blood. That is what this shows that how CRISPR is a powerful tool to control the not only virus but also plant pathogen, as well as also the controlling or remediation of the pollutants in our environment. So FRP1 knockout mutants through CRISPR case develop into adult mosquitoes showed profound suppression of infection with both human and rodent malaria parasites at the oocyte and sporozoite stages. They have observed that, and FRP1 inactivation also resulted in fitness cost. But nevertheless, that there may be physically the Anopheles that mosquito was not physically sound or healthy as wild type. They found that there is significant blood feeding propensity, fecundity, and egg machine hatching rate. They found in case of genotype or in Anopheles, which was you know which in which the FRP gene was inactivated in the embryo itself. So it has, although we aim for a positive uh, uh, effect, but there may be another associated effect also, like we have seen here in case of Anopheles mosquito. So now CRISPR is not only used for silencing of gene or editing of genes or mutation of genes, but it can also be used for identification of specific strain of the coronavirus. And this Broughton and his colleagues in 2020 they have used, which is published in Nature Biotechnology, that they have developed a rapid method where you know our nowadays detection is by rapid method or qRT-PCR currently we are doing, but there is more easy development of rapid method, easy to implement, and accurate CRISPR Cas12 based lateral flow assay for detection of corona from SARS-CoV-2 from respiratory swab RNA extracts. They have designed and more easily use this technique to detect from the swab samples of human being in United States. And they found that this is more easier compared to RT-PCR with 95% positive predictive agreement and 100% negative predictive agreement, saying that it is more accurate and less time consuming compared to RT-PCR, which also we see here. So for that, what they have done is that they have enveloped a gene. They have developed a guide RNA, because we know that CRISPR has to have two things. Guide RNA has to be there along with Cas12, which take to Cas12 nucleus to recognition site, and degrade or cast this one. What they do here is that with the fluorescence that dye, coincide collateral. You take the samples, and here is a isothermal, in the sense that normal room temperature. You mix the reaction with this different different guide RNAs that have designed from the nucleoprotein gene as well as envelope protein gene. Then different guide RNA being designed based on the CRISPR sequences present in these genes, and they found that on testing that this testing is through color observation, where they have fluorescence tag is when that that CRISPR along with the guide RNA Cas12 cluster presence of this virus, then it also caused single stranded DNA, which is a probe. Then it produces color, and we can detect that there is color, means this uh, virus is there in the swab sample. That's why they can do in the 40 minutes compared to the 2 days, almost 24 hours we need for QRT PCR starting from RNA extraction. Here we don't need to extract the RNA. You just take swab sample and just do the mixing and see the color reaction like this. Here with different genes combination or guide RNA combination, they found positive and negative based on the presence or absence of the guide RNA or Cas12 cutting site of the uh, of the CRISPR complex. So they have compared their method of development along with the that Center for Disease Communicable Disease that with the WHO report. They found that this is more time consuming or assay reaction time. We need qPCR almost 2 hours, but here 30 to 40 minutes entire process can be you know uh, completed. If we took that RNA also, that swab sample also, because our swab contains RNA, because it produces lots of RNA. That corona is SARS-CoV-2 is a RNA virus. So we take the swab sample, we can get the RNA itself, and they amplify in isothermal condition with the reaction mix, and we find that if there is color or breaking or cutting down of the that RNA, then we get color, fluorescence color. That's why it is detected as a positive. So they say that this CRISPR technology is not only being can be used for silencing or editing of the genes, but it can also be used for detection of particular strain or species of virus or any other organisms. So if you look at that, how do you identify strains in generally, we identify strains by DNA barcoding. Now you know that every mall or shopping complex, if you go, or any shop you go, there is a barcode, means specific unique barcode is given. Likewise in animal or plants or fungus, we can have different types of barcoding. Here in DNA barcoding, what we do is that small fragment of DNA, which is universal, conjugate in DNA is being amplified from 250 to 500 base pair. After extraction of DNA, we sequence it, collect the sample, extract the DNA, amplify it, and go for sequencing. Then we have what exactly this species is there in the complex of your sample mixer, we can identify. But this can identify at the species level, but from here we need here also around 40 to 24 to 48 hours to get all that, depending on the availability of the facilities you have. Here people have reported that identification of what died in these animals eat brown bear and golden mamu. From their stool sample analysis, they found that these are grass species. They are being they have eat, and that's why that is very important in case of biological conservation or biodiversity conservation. What species this animal eats, by looking into the diet pattern, we can conserve those uh, grass species, so that there will be more shortage of food for them, and they don't die. So what is actually DNA barcode is that DNA barcode are in case of plants, it is chlor plus DNA, that the RBCL or METK or non-coding special DNA. This is a one, and the internal transcribe special regions of nuclear ribosomal DNA that are being used, which is around around 450 base pair length, or depending on the species. But the primary side that we have to do PCR amplification and sequences. But in case of animal, including insects, what is mostly used is cytochrome C oxidase 1 gene. In case of fungus, we use this internal spacer DNA. So what we do is that not only those DNA barcoding can be used with the help of CRISPR technology. We can use this identification of species being or strain being present in the sample in a very short period of time, because DNA barcoding takes we have to take the sample, come to the laboratory, then extract the DNA, then amplify, then sequence, and all these things. But CRISPR technology, using the environmental sample, we can do at the side of the sample collection only 42 2 hours maximum we need for detection of particular species of any of the strain. That if you want to identify virus out of many mixture, you can identify the CRISPR technology once we know the sequences. Here in Worldwide Fund in Nature in 2018, they have reported that Planet Index reported an overall decline of 60% of wildlife population sizes since 1970, and rising to 83% of freshwater organisms. There is urgency for conservations to monitor biodiversity and track invasive species from organic material that they leave behind. It is a we need to, and not only CRISPR technology is being used for editing or silencing of gene, but it can also be used for identification of species, and being proposed by here in this William and co-worker. Williams and his colleagues in 2019 reported the application of CRISPR-Cas for single species identification from environmental DNA. That from you collect the environmental DNA, then mix the isothermal PCR and mixer tube. You have CRISPR DNA or CRISPR cassette or CRISPR reaction. You have with you, and room temperature. You can at the site of collection. You can use this one, and based on the signal as already shown in previous slide, if there is color fluorescence light coming or color changes there, we find that this is very sequence specific. That's a, we can detect that one. The method of coupling isothermal amplification to CRISPR to a detection utilizes the collateral cleavage activity of Cas12a ribonuclease guided by a specific single CRISPR RNA. So whenever we find that conserve or the target DNA in particular environmental sample, it cleaves, and as already it is labeled with probe, and that uh, RNA also single-stranded DNA also contains fluorescence, that if you cut it fluoresce or give color, that's why we can identify species. It was published in the in 2019 by Williams and colleagues. So here, what they do is single temperature of 37 degree, which is general normal room temperature, with signal detection at this is nanomole. The specific assay detects a automolar 10 to the power minus 8 concentration, very less concentration. You can detect with this particular technology with rapid detection rates at 2.5 hours compared to the 2 to 3 days, or depending on availability of facility you have if you go for DNA barcoding. So with these, they have identified fish species Salmo solar and Salmo tunata. They identify a sample site, and they have specific for CRISPR CRISPR design for this this particular Salmo solar, and then they have mixed the environmental sample and filtered it, and they found that we use the isothermal in the tube. They have amplified the DNA with the reaction mixer, and after that, if incubated with a in a few hours, and at 37 degree centigrade at room temperature, as Cas12 detection, if there is target site, it binds and cleaves and gives the fluorescent color, or give different color. So to detect that this Salmo solar is there in this particular environmental sample. But since this CRISPR CRISPR is not designed for salmon truta, it was not detected here. So by doing this, they have found that accurately in very short span of time using CRISPR technology how we can detect the species also. So now, as our you know biodiversity declining day by day, there's several biodiversity or organisms life form has been vanished from the earth water surface. That's why people are thinking how to conserve and how to use different molecular technology in conserving biodiversity and the environment. As the conservation challenges increase and new approaches are needed to help combat losses in biodiversity and slow or reverse the decline of threatened species. Genome editing technology is changing the face of modern biology, facilitating applications that were unimaginable only a decade ago. The technology has the potential to make significant contribution to the fields of evolutionary biology, ecology, conservation, yet the fear of unintended consequences from designer ecosystem containing engineered organisms has steep innovation. That is what said that every technology you develop, there may be some opposite effect also. Like what we saw in the Anopheles when there is a knockdown of that fibrinogen protein 1, there's a reduction in size or something like. But if you here also in ecosystem biodiversity, if you utilize then CRISPR technology or any molecular biology techniques, then what would be the effect that has to be carefully observed and monitored and tested. So what we can do in biodiversity, conservation, how CRISPR can be helpful. It's like there are different ways to identify the species. Like we have shown in the previous slide, DNA barcoding, and we have CRISPR slide. And with CRISPR, what we can do is site-specific endonuclease. We can cut the DNA and restriction-associated DNA sequencing. We can do and sequence and find out what are the differences in the particular organisms. Like we have programmable Indian CRISPR guide RNA. You can cut with this one, and these are different ABD targeted genotyping also. As I already mentioned that CRISPR cannot be only used only silencing or editing. It can also be being used recently to identify different species in the by the in the by the identification of biological diversity quickly, so that we can specifically say that this particular species in particular ecological condition are more suitable in this maybe because of that one. So we have CRISPR target enrichment. We can amplify the CRISPR. We like it adapter. Then we can sequence and sequence pool, and you can use this. After identifying the sequences, we can design whether we need to silence or in gene specific. Also, we can do that if any gene or this particular CRISPR sequence is present in any gene. Are they involved in the adaptation of the particular plant species or organism in particular ecological condition that can be understood, and CRISPR can be detected like this. So CRISPR is being replacing all genome engineering, genome editing, or genetic engineering technology using this. So these are some of the CRISPR technology being used for ecology or conservation of science. CRISPR genome editing is being done. Like here, in case of CRISPR targeted sequencing, characterized genetic structure of population, enrichment of large genomic fragments by CRISPR fragmentation and purification, and current use sequencing large genomic regions from eukaryotic cells. We can find out what kind of CRISPR repeats are present there. Can you use for modifying the genetic constitution, which is more amenable, more profitable, and more advantageous to particular organisms that can be found out from here, apart from identifying the particular species here. Then we can see the identify polymorphism in target genomic region, deplete unwanted sequences from NGS data. All these are some of the application, approach, current use, and references of CRISPR technology in case of in ecology and conservation science. So basically, what we can do here is that we can find genetic barcode in non-coding region of the genome, A, B, C, D, CRISPR barcode in amplify in different individuals. We find that if there is barcode, we can use and identify the organisms. Apart from this identification, we need to nowadays we have lots of contamination of soil because of excess of use of fertilizer, excess of use of pesticide or chemicals, or herbicide in agricultural field, or because of industrial area where lots of industrial spills and others are being done and degraded. That's why we need to identify any organism which uh, uh, will be able to remove or mitigate the soil erosion, maintain the biodiversity, sequester carbon. Like we need to identify algae, for example, carbon, where there will be using that carbon dioxide for production of carbohydrate, like in plants and algae, photosynthetic cyanobacteria, increase biomass production. Then if there is more production of photosynthesis, we get more production of synthesis of carbohydrate in plants or algae, restoration of natural flora and fauna. If environment is fine, that flora and fauna can be used, and we have to identify some of the organisms, bacteria, plants, or animal species which can thrive and reclaim the soil or the forest area, also erosion, and then bring back the ecology there. And eco-friendly, solar energy driven, cost effective, all are need to be identified, and people are trying to use for this through molecular biology, especially genome engineering or genome editing. Can you use this technology for all these processes by identifying organisms which are natively found in polluted area, in terms of hot spring, in terms of saline or alkaline soils, or any of the places where there is erosion, or there is habitat where normal survival of most of the organisms are not found. So phytoextraction occurs in roots where contaminants are absorbed and transported in various harvestable plants that are converted to energy, and on burn and metal can be recycled from the ash. These are different definitions of how we can extract or reclaim the soil, or the any area, or or water bodies, by using different organisms like plants, algae, or bacteria. Can you remove? Can you degrade? Can you use more of the carbon dioxide being found, or methane gas being found in the environment? Can there be any organism which can use the most of the greenhouse gases, or reduce the greenhouse gases? That has to be through molecular biology. We need to identify the genome sequences or genes. Then only we can edit those and design organism, for example, algae or plants or animals which can be useful and can survive in those areas. So engineering plant growth promoting microorganisms through gene editing system biology, and it has been found that uh, you, you know uh, plant growth promoting microorganisms also found at the where if you grow the plants or crop plants in the affected areas, like polluted area, then can you use some of the microorganism along with that? And we need to identify what are those genes being found, which are helping in survival of synergistic relation of the plant with the microbe, and helping growing of the plant. That has to be identified. We have to sequence the whole genome. Then we have to identify protein. We have to identify what is the metabolic biosynthesis inside your cell, and then correlate with their proteome, transcriptome, and metabolome. Identify some of the genes which may be involving in all those phenotypic trait, or giving plants survive in its particular environment. These are some of the plants or organisms, a list of genes identified from microbes in their host to improve phytoremediation. First plan is this, for example, auto declination gene. This is pseudomonas aeruginosa. Target is common. This is the plan. Then enzyme encode metabolized chlorbenzoic acid. These are being used to mitigation of the uh, mitigation of the all the environmental hazards, pollutants, or some along with the plants. And people are trying to identify what are the genes that are responsible for those tolerant behavior contributing this one. So integrated omics genome editing tools for phytoremediation. We have this is a transcriptome, genome, proteome, phenomics, all coming to understand the function of genes, or if not, we can just identify gene sequence and with CRISPR-Cas or any other molecular genetic engineering, gene editing technology. We can see what is the effect of silencing of the gene and see how the abrogation of the or removal of the particular chemical compound in the soil can be takes place. So this is as simple as that. Genomic, how CRISPR can be used. Genomic DNA, target sequences, guide RNA. We find it and see that particular organism gene. If you find plan is there, then metal ligands, if help in metal transport and something like it help, then we can see this gene. If we can enhance or we can silence these genes, which helps in removal of metals or phytoremediation. So it is very helpful. Examples of few plant species involve phytoremediation, environmental pollution. We can find out phytostimulation. Then we have phytoextraction. All are there. So we have biofuel production also. There are different organisms which can be used for biofuel production, like acetogen, chemoautotrope, photoautotrope, methanotropes. All these are bacteria that can be used for biometal or can be used for metabolic engineering to produce biofuel and chemicals. So these are some of the example targeted metabolic efforts towards selected platform chemicals, biofuels with most prominency on such of the. These are the different with butanol acetate. Many research group have tried to use those organisms to produce bioethanol or biodiesel. So this is algae that Clostridium. This Clostridium involves in the greenhouse gas emission abatement can be now be achieved through gas fermentation using acidogenic microbes for the low production of carbon fuels and chemicals such as Clostridium. This is bacteria that they have reported, and they have studied the mutagenesis study also. What mutagenesis, can you use the mutagenesis to grow to silence some of the gene and see how it is affecting the phenotype, because this Xenococcus elongatus, these also for photosynthetic microbes are of considerable interest for applications in carbon sequestration, photosynthesis, production of fuels, and biosynthesis of other valuable chemicals such as pharmaceuticals. They grow on carbon dioxide and sunlight alone. This reduces greenhouse gas emission and decreases dependence on petroleum-based products. They're the evolutionary ancestor of plastics. That's why they're being used. And if you can, edit the genome, develop a protocol which can enhance the, growing and utilization of carbon dioxide for photosynthesis, then it will be very good in biofuel production. So gene editing for climate change, biological solution for curbing greenhouse emission. I'll take just only 5 minutes more. So this ITIF, Information Technology Innovation Foundation, they have given some of the until 2050. They have target what way we can reduce the greenhouse gas emission, reduce food loss and waste, phase out crop-based biofuels, increase crop yields, plant existing crop plants and more frequently, increase faster, improve aquaculture, reduce enteric fermentation. All this will reduce to by 2050. If you these processes are being followed, these steps are being followed, they feel that greenhouse can be reduced to gas can be reduced to a certain extent. So in plants generally, there are so many development, first hybridization and how to develop plant variety which can be used for, for example, there are so many plants which can, be used for phytoremediation, removal of pollutants and others, including rice. That I'm coming to the next slide. That, but before that, plant improvement have been different conventional methods, and only recently, until 20, early, 2011 and 2012, the class, CRISPR technology have developed. That's why without breeding and going for, developing crossing and combining genotype from different individuals, we can directly go for at the genome level and see that, you know, yeah, how to use this without wasting lots of time. So what we can do is globally, reducing food loss and waste. This is also causing environmental, hazard. Globally, 1/3 of food grown lost per year, as per report of Food and Agriculture Organization. And that decomposition of wasted food and the fossil fuels used to produce and save it, it makes the single largest component of agricultural greenhouse emission, as much as 1.9 million tons of carbon dioxide equivalent warming potential to the per year. The first gene-edited food, food product to reach the market was silo high olive soybean oil for food service application by biotech startup Calyx. Uh, Calyx, this is the company that produces all high oleic acid soybean company, producing soybean. Gene-edited, crop. It helps in, making the oil quality survive more than 1 week. Generally, what it does is, oil generally, if you
Dr. Nirala,
yes,
your voice is becoming low. Can you hold up?
yeah, is it okay? Hello, okay. So this high oil, soybean oil is being developed by by CRISPR technology and being, developed by Calyx company, and it is good for heart, because it has high oleic acid content, and it is being used and good for, deep frying of any, potato, any other, vegetables, and it can be used more than 1 week. But generally, currently we can use any natural, oil we can use up to maximum of 1 week. So we have to look for weed control by, by CRISPR technology, any of the molecular technology. Can you control? Finishing almost 2-3 minutes. So herbicide tolerant, you can use aquaculture in the sense that if you use more aquaculture, biodiversity still be conserved. There will be no damage of environment, as well as, reducing dependent on the plant product which produces, more of, like for example, rice produces more carbon dioxide or due to fermentation of one area, Mars area. Reducing ruminant emission, it has been shown that 1.4 billion cattle comprise the second largest source of agricultural green emission. So in the through, genetic engineering, this group, Su et al. Has found that expression of barley SUSIBA2 transcription factor, they found that high starch content, low methane production of this rice. And if you look at here, that root is somewhat less, and production of grain is more, having high starch, as well as they've studied different emission of, methane also. They found that methane is less, and they have, it produces more of, source to sink, and more grain, high yield, less methane. So we have, now Danish, they have, Danish government, Danish team, they are finding of, found, developed through gene editing super grass to increase cow milk production and produce less methane emission. We have Swiss grass, that is biofuel being used, and being, scientists are being trying to understand what genes can be, silenced, lots of processing is there through gene editing for biofuel production, improving photosynthesis, enhance growth, protecting, restoring ecosystem. All are like this. If you want to, because fertilizer is being, percolated down the grain, if you can develop more roots, it happens like this. Then this experiment says that by using this XP1A, XP1B, they have removed the RDX contaminated soils in USA, and this is the scientists, they are being used in sweetgrass by gene editing technology, genetic engineering. We have adapted traits in forestry. If you can identify any of the trait in forestry which can be used for replenishing or replanting of the forestry, and some of the uh, genes that genes and species that are being used for finding out the trees in the forest and environment by genetic technology. And this is one where cucumber mosaic virus resistant we develop. And this is the last slide, few slide, that we can say that because of gene editing technology is being widely accepted for for being aimed to produce food sufficient in stress environment conditions in, like in case of Africa cassava, recently in banana, CRISPR edited one banana genotype has been developed in Africa, where it is gentleman's will resistant. It is giving more of yield due to non-susceptibility because of edited genes. There are so many reports where climate change we can use in CRISPR gene editing technology, producing of more oil in case of algal fermentation, identification of phenotype, or suppressing of genes in case of anopheles, and increasing sturdiness or detection of phytogen. All are being used gene edited technology for easily use in the changing climatic conditions. So this is the microalgae also. So with this, I conclude that not only that not only editing gene, once we identify gene or genome sequences, we can design a CRISPR technology, and any gene can be edited for any trait, for for enhancing the performance of plants or animals or any other organism, which will be very beneficial for human utilization of by human being. And thank you so much, Professor Borai, once again, at Center for Environment. Now I will conclude my talk here. Anything that you can ask?
Thank you, sir, for this wonderful presentation, sir. This is Adhiraj Nath, Professor Utul Bora, sir, student. So now,
Yeah.
now I would like to ask the participants if they have any question to ask, they can post it on the chat box. In the meanwhile, let me thank you for this wonderful lecture. It was so informative. So I hope everyone learned a lot from it. And thank you for finding out time during this hard. You are going through a problem. Your family, your son is not well. We understand that, and we really thank you for finding time for this presentation. So I think there are some questions here. So one question is from Raj Ved. So he asks, any role of gene editing in controlling carbon emissions? Sir, can you read the question?
Yeah, that is what, like for example, rice, and that if you can utilize more photosynthetic carbon emission is done generally by for example, fossils also, as well as greenhouse gas emission also. People are trying to use gene editing technology where you can use, like in case of rice, they have produced more of diverting the source from the root zone or source to the sink zone, where more grain is being produced, and more photosynthetic efficient, where we can use more of the carbon dioxide and produce less carbon dioxide and produce more oxygen. That's why people are trying to utilize cyanobacteria plants to reduce the methane as well as carbon dioxide emission in the atmosphere. So hopefully we may get some reduction in near future.
Okay, sir. Okay, so, sir, there is another question from Pinku Mani Talukdar. So why GMO crops are resisted in India for launching? And what is the future of such crops in coming time?
Well, GMO, people believe that one is that it may go through wild, wild type or natural type. It hybridization. It may be super resistant to diseases or something. Maybe there. And one is that people are skeptical about the markers in like antibiotic resistance genes. We earlier genetic engineering technology, people used to have antibiotic resistant gene as a marker, selective marker. If it goes with the food, that people think that it may affect our health and benefit. That's why. Otherwise, if you look at corn, soybean, and cotton in USA, and whatever corn oil, soybean oil we import from USA, we get all a GMO. But very tragic part is that we don't allow to grow those crops in eatable, except brinjal. That in Bangladesh, we have not allowed any of the transgenic in India, and we have ready mustard also. Mustard also being allowed to grow in India, but we are importing $1 million oil from abroad. This is the tragic part of India.
Dr. Ramchiary, you may put your video on.
Hello.
Sir, you're not audible properly.
And
Yeah, can you hear?
But also your voice is breaking up. Hello?
Can you hear?
Yeah, I can hear you. So now of your presentation you can put on your video.
Hello? Can you hear?
Yeah, fine. I can hear you.
Okay. Okay. So anything more?
So there is another question for you, I think. This question is from Bushra Khatun. So algae is being mass produced to replace plastic as it's biodegradable. But how would the byproduct of algae degradation impact environment?
Biodegradation of?
Algae is being mass produced to replace plastic as it is
Right.
biodegradable. But how would the byproduct of algae biodegradation impact environment?
No, this is biodegradable, so it doesn't impact the environment like plastic. So in fact, people are trying to use bacteria against this plastic which are currently being available. So it is environmental friendly that biodegradable plastics are environmental friendly, and people trying to use from cassava carbohydrate and other algae also, from plants also, that can you design some bioplastic like this.
So there is a question for Pranav Kumar Nath. Was that answered? I missed actually. There are some connectivity problems here.
What
How
was that?
is the prospect for increased? Pranav Nath has asked prospect for increased biomass production in energy plantation. I think that was answered, right?
Energy.
No, sir.
He want to, as I can understand, do genome engineering for increasing the biomass production. So I can also answer this, Pranav. Yeah, it is possible. So, and then your next part is Dr. Pranav. Can you hear me? So he's asking in continuation. Also, kindly let me know, will there be regulation to cultivation in field? Yeah, definitely there will be cultivation, or there will be regulation.
Yeah,
Right.
there will be.
There will be regulation. Yeah.
Biomass production, I have just given in the right.
There is another question for you, Dr. Nirala. Can we get feasible detection of plant pathogen with genome engineering techniques, especially poor farmers of India?
Yeah, if you can find a CRISPR, sequence like isothermal that I have just shown in case of salmon and coronavirus, that if the technology becomes available, definitely you can on-site field, only 2 hours of we can do that detection of pathogens. But we have to design that first, get the sequence and design.
Yes, and some systems people are trying to develop of contaminants or attack by pathogens. Whenever the pathogen or the contaminants will be there in the environment or in the host,
yes,
it will elicit such a response. Some kind of fluorescence signal will come out.
Yes.
Okay.
Yeah.
So these are some of the known strategies.
Yeah, biosensing. Yeah, biosensing. Yeah, people are trying to
Yes.
develop. Yeah.
Yes. Dinesh Pratap is telling thank you. So, Dr. Nirala, thank you so much. And it was such an informative lecture and very extensive lecture, and you have covered so much. And in spite of your difficulties, I hope your son is okay now. You had taken all the trouble to deliver such an valuable lecture. So we look forward to one more lecture from you. But I understand you are under a lot of problems. So I see a lot of appreciative comments for you, thanking you for the lectures, for the lecture in the chat box. So
thank you all.
any other questions are there?
Yeah.
Participants can still ask. Okay, so I think it is lunchtime, and then we will have another lecture after this. So I did take this opportunity to thank you formally for delivering this lecture and for consenting to be a panelist. So I once again request the participants to press the Y button and give Professor Nirala Ramchiary a round of applause. Thank you. Dr. Nirala, if you'd like to say something in concluding, mic is yours.
Yeah. I would like to thank all the participants, because yesterday, despite your schedule time, I have failed you in losing time. And today I tried my best, but I would be very happy if you give your comments. And then lastly, I thank Professor Bora and the Center for Environment, as well as Adidas and colleagues, for taking that much of trouble to get me in and giving this opportunity to share some of my observation in the field of genome engineering and its application all in different organisms to develop product which will be beneficial to human being. Thank you all. Thank you so much.
Okay, dear participants, we will have our next session at around starting at 2:30 p.m. Thank you. Please be there and have lunch break now.
Okay. Thank you.
We'll have